Hello students, hope all are staying safe at home. Today we are going to discuss about yet another deleted chapter during the last academic year, that is mineral nutrition. I will not be discussing the whole, I will just give you a bird's eye view of the chapter. Okay. So mineral nutrition, the first term what you have to notice, what is hydroponics. Okay. So hydroponics means what? Hydroponics is a method in which the plants are grown in a nutrient solution where only in water. So we are taking only water. The plants are usually grown where it is the soil, right? But in hydroponics, soils are not used. Instead of that, what we are using? We are using water. So in a medium containing water, what we are doing is that I am culturing the plants. So imagine this is a beaker which is containing water. In that I am going to cultivate the plants. Okay, I am going allowing the plants to grow in water. So essential nutrients, what and all is needed, that we are putting it in this water, and then we are culturing plants in the water. So this sort of technique we call it as what? Hydroponics, right? Now this hydroponics is very important because by understanding this hydroponics method, we can understand which is essential for the growth of the plant, which nutrient, and which nutrient is not essential. Like for example, I am taking sodium and just trying whether just seeing that plant is growing properly or not. In case the plant is growing properly and the growth is becoming more, means that sodium is needed. And some other element I am taking and putting, and I am saying there is no considerable change in the plant, means that nutrient is not that important. So I can come to know about these things. Okay. So these is a hydroponic is a technique nowadays richly available. You can find wherein they are using water as a medium for the culturing of the plants. Okay. Now the mineral nutrition, in this chapter we are going to discuss about the different minerals which are needed for the growth of the plants. So these minerals which are needed, we classify them as essential elements. Okay. So you know that more than 60 elements are there, but out of those only 17 elements has been considered as essential elements, because the 17 elements is directly involved in the growth of the plant. Okay. So that's why it is called as what? Essential elements. So how I am classifying whether one element is essential or not? Like for example, carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, magnesium are very much essential for the plant. When do I call this as an essential element? We call this as an essential element if it is fulfilling some criteria. Now what are the criteria that this essential elements have to fulfill? The first criteria is that the elements must be absolutely necessary for supporting normal growth, which means that the element should be there. Only if the element is there, then the plant will be growing in its own way. Okay. So if this element is absent, in case if this element is absent, then automatically that life cycle or the seed development will be disturbed, which means that that element is very much essential for the plant. Okay. That is number one. Second is that the requirement of the element must be specific and not replaceable by another element. Means I am having element A. I applying this element. Instead of this element A, if I apply one another more element called as element B, then the plant is growing in the same way. They are having fulfilling the criteria of A itself. Means I cannot call A as an essential element. So if I am applying A, then only the plant is having some changes, which when I put some other elements, that change cannot be fulfilled. Means that change is not happening. Okay. So then I call that one as what? The essential elements. Okay. So deficiency of any one of the element cannot be meant by supplying with other elements. So if you are just substituting with something else, if the deficiency is fulfilled, then I cannot call that element as what? The essential element. So the element must be very specific and it cannot be replaced by anybody else. Like for example, one student is very specific. That student cannot be replaced by some other student. So the same thing is applicable here. Then that is an essential student, or that is the essential element in this case. Okay. And the last criteria is that the element must be directly involved in the metabolism of the plant. Metabolism, you know what is metabolism? There are two mechanisms. One is catabolism and another one is anabolism, isn't it? So for this catabolism as well as for this anabolism process, that is for either breakdown like respiration or photosynthesis, anabolism, in these reactions this element should play a major role. Then I call this element as what? An essential element. So 3 criteria has to be fulfilled. 1, it must be absolutely necessary. Second, it should be very specific and should not be replaced by someone else. And the third one is that it is directly involved in the metabolism of the plant. Okay. So these are the things which are fulfilling the criteria for essentiality of the element. Now moving on to classification of mineral nutrients. Mineral nutrients are classified into 2 categories. Okay. So what are the 2 categories? One is the micronutrient and another one is the macronutrient. So what is macronutrient? As the name suggests, macro means what? Which is needed in high amount. We call it as what? The macro. Micro means what? The which is needed in very less amount. We call it as the micro. So these macronutrients are needed in more amount. So which are the macronutrients which are needed in more amount? It's carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium and magnesium. And what are micronutrients? Minor elements, that is very less needed. Okay. So what are they? But it is needed. Less, but it is needed. It cannot be substituted by anyone. So essential only, right? Now this micronutrients, when you are saying, we can also call it as trace elements. They are iron, manganese, copper, molybdenum, zinc, boron, chlorine and nickel. Okay. So these are the elements which are needed in micro concentration, that is, it is needed in very less amount or trace amount. Okay. So these are the things coming under what? The beneficial elements. Now the next topic what we are going to discuss is deficiency symptoms. Okay. So what do you mean by the term deficiency symptoms? Deficiency symptom is that when you are seeing, actually what is happening is that the plant is exhibiting some of these symptoms which is showing that the plant is having some deficiency in some of the elements, because of which that symptoms are being shown. Like for example, there is an irregular growth in some part of the plant, or there is a different colored nature they are developing. Okay. They are turning yellow soon. Okay. Yellowing is taking place, or they are dying soon. Stunted growth. Okay. So these are some of the deficiency symptoms which are noted in the plants. Okay. So an element, when we call it as it's deficient, actually when I call an element is deficient, when it is below the critical concentration. What do you mean by critical concentration then? So what is critical concentration? The concentration of the essential element below which the plant growth is retarded is called as what? Critical concentration. Like for example, sodium is needed 100 mg means, 10 mg means, then this is the critical concentration. If it is having only 9 mg or if it is having only 8 mg, then they will be having some deficiency symptoms. So how much is needed? Minimum how much is needed? It's 10 mg. As for example I am saying. Okay. So then I call this 10 mg as what? The critical concentration, below which the plant is not growing properly, the plant growth is retarded. Then that concentration of the mineral nutrient or the essential element, we call it as what? We call them as the critical concentration. Below this critical concentration, plant will be exhibiting some deficiency symptoms. Okay. So what are the deficiency symptoms? Some of the deficiency symptoms are listed. Chlorosis. Okay. So what is chlorosis? Chlorosis means when you are seeing the green colored part is means it's not becoming green colored things, when you are seeing that is turning somewhat differently colored. So that necrosis, you can find tumor like growth in the plant cells, that is necrosis. Inhibition of the cell division is not taking place, the plant is not growing in height, the roots are not growing, the stem is not growing, the leaves are not growing properly because of deficiency in what? The cell division. Or the flowering is not taking place, the plant is growing but the flowers are not seen. So these are the deficiency symptoms of some of the deficiency symptoms which the plant can represent. Okay. Now moving on to the next topic is role of nutrient elements. So what are the different role of nutrient elements? So these role of nutrient elements we have already discussed. Okay, in your lower classes. Again, nutrient elements roles I am not going to discuss in detail, just I have be showing you this one: nitrogen, phosphorus, potassium. Okay, thereafter you can find calcium, magnesium, sulfur, right? Then iron, manganese, zinc, copper, boron, molybdenum and chlorine. Okay, so these things, if it's not present. When you are seeing these nutrients, what are their, when you are seeing the topic, the first one example I will tell you. Elements, nitrogen is given, where it is obtained as, okay, how the plant is getting. It is mainly obtained as nitrites and nitrates, as salts from the soil, okay, we are taking it, or as ammonia from the water. When you are seeing the water plants, they can take in. So like that, the new nitrogen is reaching the plant. And what is the role of function? How it is playing a major role? Required by all parts of a plant. Nitrogen, when you are saying, because it is a component of protein, automatically it will be required by all parts of the plant. Particularly it is needed in the meristematic tissue where the cells are dividing actively. So there you can find they are particularly needed, okay. And they are the major constituents of what? Proteins, nucleic acid. For making nucleic acid, nitrogen is needed. For vitamins, for hormones, making hormones, for proteins, everything what is needed, nitrogen is needed. And when this nitrogen is not formed in required quantity, the plant is not getting, then what are the deficiency symptoms? First is chlorosis, second one is inhibition of cell division, and third one is what? The delays, they are delaying the flowering. So like that, for each and every nutrient element you can find how it is obtained, what is its role, and what are the deficiency symptoms has been discussed in these tabular column. So it will be easy for you to just go through, okay. So you can go through, I am not explaining each and every, it will take time. Now important part of the chapter is metabolism of nitrogen, okay. So nitrogen metabolism, what is the role of nitrogen, how it is getting plants, how it is getting into the plants, and how it is helping in the formation of amino acids which are making the protein. And you know, proteins are the building blocks which are helping in the building of the plant, isn't it? So metabolism of nitrogen, the first thing what we are going to discuss is nitrogen cycle. Nitrogen cycle having 4 steps. What are they? One is your nitrogen fixation, second one is your ammonification, third one is your nitrification, and the fourth one is your denitrification. So one by one we will be seeing. Atmospheric nitrogen which is present in the atmosphere is fixed by 3 methods. One is biological nitrogen fixation, wherein the living organism are playing a major role. Industries is fixing nitrogen. Electrically, when you are seeing the lightning, okay, thunderstorm lightning is helping in fixing the nitrogen, okay. So these are the various ways by which the nitrogen are fixed in the form of ammonia or in the form of nitrates. The what is happening, actually the nitrogen is getting fixed, okay. So when this ammonia, how it can also get fixed by some other process. So these nitrates, nitrites and all which is present in the soil is taken by whom? The plants, okay. So the plants are taking this nitrogen from the roots, it is going to the stems, to the leaves, okay. There what they are doing, they are synthesizing the proteins. So first amino acid they will synthesize, then the proteins, and when these plants and all die, okay, so plants are being taken by whom? The animals, isn't it? So we are taking vegetables, along with that the nitrogen is reaching us. Now this plants and animals, when they die, actually what is happening, they are buried under the soil or they are put under the soil. So what will happen? These plant and animal will be degraded by microorganisms, and the degrading biomass, when you are seeing, there are a set of bacteria called as ammonifying bacteria which help in the process called as ammonification, where these proteins of these plants are getting converted into ammonia. This process we call it as what? Ammonification. Now biological nitrogen fixation, in that also you can find directly the soil organisms converts this nitrogen into ammonia and they fix it in the soil. Now this ammonia has to be converted into nitrites and nitrates, okay. So NO2 is nitrites and NO3 is nitrates minus. So why it has to be converted? See, ammonia is not readily available for the plants. It has to be broken down into more simpler form which can be taken by the plants. So it has to be converted into what? Nitrites and nitrates, okay. So that is what is happening when you are seeing in which process this nitrogen cycle. So fourth thing we have discussed, nitrogen fixation is done, ammonification you have understood, nitrification you have understood, isn't it? So I will repeat again. So nitrogen is fixed in the form of 3 ways: biological, industrial and electrical, as ammonia or nitrites. These nitrites are taken by the plants and it is converted into plant protein, and plants are taken by animals, it is converted into animal protein. When these two die, they are being degraded, okay, and the process which is taking place is called as what? Ammonification, wherein this protein are converted into ammonia. This ammonia cannot be readily taken by the plants, so it has to be converted into nitrites and nitrates. Who is doing nitrifying bacteria? That is why the process nitrification. The last process is this nitrites and nitrates, when you are seeing, which are present in the soil, are converted by another set of bacteria called as denitrifying bacteria, which converts this nitrites and nitrates into atmospheric nitrogen, and they release it into the atmosphere. So, like that the nitrogen is reaching the atmosphere. So, this is regarding your nitrogen cycle. Now, biological nitrogen fixation in detail we are going to discuss. So, in this conversion of nitrogen into inorganic or organic usable form through living organisms, we call it as biological nitrogen fixation. Can be of two types of microbes which is carrying out this process. One is free living organism and another one is symbiotic. Free living, they live freely. Symbiotic, they live in association with some other organisms. Okay. So, symbiotic, you know, we have learned lichen, algae and fungi, isn't it? So, one as well as the two, both are mutually benefited. So, that one we call it as symbiotic. Now, free living bacteria, when you are seeing what are the different free living bacteria, free living bacteria fixers are there. They are about 30% of nitrogen is being fixed by these free living bacteria, and symbiotic bacteria are fixing about 70% of nitrogen. Okay. So, examples of these nitrogen fixers are given. Free living bacteria are Acetobacter, and examples you can just look into. Okay, Beijeringia. Free living bacteria, anaerobes, when you are seeing, one is aerobes. Aerobes means in the presence of oxygen, anaerobes means in the absence of oxygen. Cyanobacteria is what? The blue green algae, and symbiotic bacteria examples are given, you just go through. Okay. And this biological nitrogen fixation, what and all is required? A reducing agent is required, ATP is required for providing energy, and one enzyme is required. The name of the enzyme is nitrogenase, and there is a molybdenum protein complex actually is needed, and thereafter it need a compound called as leg hemoglobin. Okay. So, these are the things which are necessary for biological nitrogen fixation. So, when this bacteria, when you are seeing this rhizobium is present in the plant, they help in the formation of nodules. So, where is this rhizobium present? They are present in the root nodules of leguminous plants, that is the pulses. Okay. So, in that only you can find this rhizobium is present, and how this rhizobium is going to help in nitrogen fixation, that is what we are going to see. Okay. So, you can find in this picture it is given. So, nitrogen, these bacteria are present in the roots. Okay. They easily penetrate what? Into the roots. Okay. So, what happens when bacteria come in contact with these roots? The roots will try to curve round. Okay. And the bacterial spores will be entering into first they will enter the root hair, thereafter they will enter the epidermis. Okay. Thereafter they will enter the cortex. When they reach the cortical cells, they start to multiply and they help in the formation of this structure called as what? The nodules. So, like this rhizobium is forming root nodules where in the leguminous plants. Okay. So, root nodules is being formed, and this mature root nodules is helping in nitrogen fixation. You see, I told you there is an enzyme which is needed for nitrogen fixation, nitrogenase. So, what this nitrogenase do is that this nitrogenase is actually the enzyme which is attracting nitrogen towards its. Okay. So, this nitrogenase which helping in the attraction of the nitrogen towards it. So, this NN is the nitrogen which is coming and binding. You can find one binding site of nitrogen is present there, the nitrogen is getting binded. Okay. So, where in the root nodule you can find this process is taking place. So, when this nitrogen is going and binding, you can find this nitrogen helps in the attachment of this hydrogen. Okay. So, hydrogen goes and attach with each nitrogen. So, 2 hydrogen is going and joining. Again, next step, another 2 hydrogen will be going and joining, and the next step reduction is taking place. Another 2 hydrogen will be going and joining. So, for 1 nitrogen, how many hydrogen will go and join? Altogether 3 nitrogen will go and join, and they result in the formation of what? They result in the formation of ammonia. So, how many, an enzyme nitrogenase, how many ammonia they can produce? They can produce 2 ammonias. Okay. So, there is another one compound which we call it as leg hemoglobin. I told you what is actually they are doing is that they are oxygen scavengers, means this nitrogen genase enzyme will function only in the absence of oxygen. If oxygen is present, then this activity will not take place. So, what this leg hemoglobin will do, they will absorb all the oxygen towards itself, so that the process continues. Okay. So, what is the process taking place? I have told you what is the process. The process is that the nitrogenase enzyme attracts the nitrogen towards its nitrogen molecule, and they helps in the 3 consecutive reduction division wherein hydrogen is added at each step, and they help in the release of these 2 ammonia. Okay. What is the next process? What is happening to this ammonia which is formed in the root nodules? This ammonia, when you are seeing, it has to be converted into protein. Okay. So for forming proteins, you know, it is made up of amino acids. So the ammonia has to be converted into amino acid. Okay. So the first process in means making of amino acid is that reductive amination. Okay. So in what is happening in this? Ammonia is reacting with a compound called as alpha kletoglutaric acid is reacting with this ammonia which is formed with the help of the reducing power I told you which is needed. NADH2 is the reducing power. They are forming what? Glutamic acid. What they are forming? Glutamic acid. Okay. So this glutamate is one amino acid. This is the amino acid which is formed. This amino acid is acting as a main source which is helping in the formation of other amino acids. How? That is the next process called as what? Transamination. Okay. So in transamination, actually what is happening? Transfer of one amino group from one amino acid to the keto group of another amino acid. Actually they are transferring the groups. So this is glutamate, okay, and this is another amino acid means some other amino acid group is present. So actually what is happening is that this B, the group when you are seeing, that is getting transferred. So transamination, as I have already told you, that is not more clear. So this one you see. So when you are seeing, this is the glutamate which is formed. Okay. So what is this? This is the glutamate. So this glutamate, when you are seeing, this is the main amino acid which is formed, and there is another amino keto acid. So this is R2. Okay. R2 is any alkyl group you can find. So this R3 will be another amino acid, R4 will be another amino acid. Based on the number of carbon molecule, the amino acid name will be different amino acids are formed. Okay. So in transamination, this glutamic acid is always present and some other keto acid will be present, maybe R2, maybe R3. So based on that you can find what is happening, the rest of the amino acids are formed. So what is happening from the glutamate, this R1 is going over here and the R2 is coming over here. So I am getting another amino acid. R2 amino acid I will be getting. Now if there is a R3, then what will happen? I will be getting R3 amino acid. Like this, the different types of amino acids are produced with the help of the main amino acid. What is the main amino acid? The main amino acid is your glutamic acid. So first it is helping in the formation of this glutamic acid, glutamate or glutamic acid. Okay. With the help of this glutamic acid, the next process, transamination, is taking place, wherein the other amino acids are getting synthesized. Okay. So some other keto acids comes with R2, R3. Based on R2, R3, R4, whatever the alkyl group it is, I am getting different amino acids by just transferring the groups. Okay. The functional groups from one to another just getting transferred, and I am getting different amino acids. So this process we call it as what? Transamination. Okay. So with this, the chapter comes to an end. Hope you would have understood. Thank you.